The goal of this first lecture is to give you an introduction to the discipline of evolutionary developmental biology, give you a few basic definitions. We'll also talk about some big examples of some of the questions that can be addressed using EvoDevo. We'll just kind of give you an outline and we'll talk about them in more detail later in the semester. Okay, so basically, when we're talking about evolutionary developmental biology, we are trying to fully understand the process of going from the molecular level, that is DNA, which is the genetic code for all life on Earth, to the morphological and behavioral and tissues, organs, all of that complexity that we have in fully formed organisms. Now, morphological complexity reaches its peak in the animals, the metazoa. And so for this course and most of EvoDevo focuses on animals. We will have a very brief section at the end where we look at plants. Plants do have some diversity, but really when we're talking about morphological diversity, it is far and away most advanced and complex in the metazoa or the animals. So just be aware, metazoa is a scientific name. Animalia sometimes is used, but metazoa is more proper. And we'll use the term animals also as a more everyday common name for that group. Now, evolutionary developmental biology is a relatively new field. We live in an age where we have new disciplines of biology that are coming online quite frequently. So maybe not as new as some of the newer molecular fields, but still uses many of those techniques and is continuing to advance and to become more and more of a comprehensive discipline. It's commonly called EvoDevo for short. Some people will say evolutionary biology or developmental biology. Really, it is a combination of the two of those. And so we'll make sure you have clear definition of what evolution is and what developmental biology is and how we can bridge the gap between those two. As mentioned in the orientation video, we will be having readings from the textbook here, from DNA to Diversity. It's a little bit different than most textbooks. It's a little shorter. It's fairly approachable, good read. I highly recommend you have it. We'll go over all of the material that you need to know in lectures and other material posted here on Blackboard, but the book will be a very good resource for you, so I highly recommend that you get a copy of it. So let's quickly define evolution. Most of you have had an evolution class or at least some amount of evolution in previous biology courses. Evolution is just very simply change in a population over time. And of course, when we're talking about population, we're talking about a group of living organisms. You can replace that word populations for species. There are some slight distinctions there, but for our purposes, that will work fine. So if you want to say change in a species over time, that works great. And there are lots of misconceptions about evolution. I hope that you have a fairly solid grounding in evolution. You're fairly comfortable with that. We will be talking about and reviewing some of the concepts that are taught in the evolution class. I teach the evolution class here on campus. Also, there are other instructors that do it. And so if you've had that class, you will get a bit of review, but I have learned from past experience that it's important that we review those and go over those and maybe get rid of some of the misconceptions. This is just kind of a little funny approach and a take on evolution. Realize that all species are constantly changing and evolving, some faster than others. Maybe it would be more appropriate for this joke. As a scientist, I sometimes nitpick a little bit, but might be more appropriate to say that the hamster would not eventually evolve into a dog, but potentially could evolve and change into organisms. And this is, again, a population could change, not the hamster itself, but its descendants in many, many generations could change into something that was more dog-like, that looked quite a lot like a dog. It would not be a dog because each population is unique and distinct. And that's part of what we're going to be delving into as we look at the genetic makeup of organisms and how that genetic makeup translates into morphological complexity. So for many hundreds of years, scientists have studied development. There have been various mores, rules against looking at human tissues and human cadavers, but even going back to Da Vinci, we had a fairly good understanding of some of the developmental processes that were going on, going back hundreds of years. Coming up into modern biology, as people began to gather new tools like microscopes, modern dissection, surgical techniques, um, we began to understand the physical large processes very, very well. But near the end of the 19th century, we kind of reached this stalemate, if you will, or a dead end, where we had done about as much as we could with the limitation of the tools and without an understanding of what the underlying molecular forces were that led to that morphological diversity. And so classical development is still a component of EvoDevo. It's important that we understand physically what is going on, but really we're kind of looking at a more deeper level than that when we're talking about evolutionary developmental biology. We're looking at the genes and how the genes lead to this more complex form. So we could do that with humans. We can do it with any animal. This is a diagram of I believe this is a cricket. And so how do we go from a single cell, fertilized cell, to a small cluster of cells, to a little bit more complex, more and more complex, till finally, going through this entire process, we get a fully formed embryo ready to be born and begin an active life of its own. And so that's the process that we're going to be looking at. Now, arthropods, like this cricket, are a really great group to work with. They're very tractable, meaning that the logistics are not too extreme when we're going to be using them as model organisms. And so much of the original work and much of the ongoing work today is done with arthropods. And so we'll focus a lot on arthropods, but we'll also branch out to other types of animals and kind of span the animal kingdom, at least the common phyla, the major groups. So I'm going to start with one example of the types of questions. This is one of the big questions that's still being worked on for the types of questions that can be addressed using the discipline of EvoDevo. So, in very simple terms, where did wings come from? And so to do this, we need to understand a little bit about the diversity and complexity of wings that are present in the animal kingdom. So let's look at this and we'll answer this question just in part, and then we will put a pin in it and leave it for further investigation at a later portion of the semester. So how many times have wings evolved? Now, when I say wings, I mean fully formed wings that provide lift and allow for sustained flight. I'm not talking about a structure that you can hold out and glide like a flying fish. There are some frogs that use the pads of their feet and can glide as they fall out of a tree in the rainforest. But we're not talking about gliding. We're talking about fully formed flight. So as we look across the animal kingdom, there are 4 clear separate instances of the origin of true flight. So think about it. What groups are they? You probably know maybe all 4 of them. So take a minute, think about it. And one of the interesting things as we answer this question is that 3 out of the 4 examples evolved in the amniotes. Amniotes are a group of tetrapod vertebrates that have a membrane around their embryo so that their eggs can either stay on land or can be held internally as the embryo develops. So wings have evolved in the birds, the bats, right, a group of mammals, and then in one other group that is now extinct but was distinct and different from these 2 groups, the pterosaurs. So these are all examples of vertebrate evolutions of true flight. The 4th example is the insect, and we'll come back to the insect in a little bit here, but let's look at this one and kind of talk about some of the things that we would need to investigate and study if we're going to begin to lay a foundation for an evolutionary developmental study of the origin of wings. So, first off, if we're going to answer this question, where did these wings come from? This is a fairly obvious answer for all 3 of these groups of organisms that separately evolved wings, right? All of them have modified their forelimb to a wing. And this brings up a really important topic that we'll delve back into later. And I'm just going to leave it open for now. And I hope you realize that this is a complex question. So if I were to ask you, is the wing of birds, the wings of bats, and the wings of pterosaurs, are they homologous structures? Now, that requires that you understand homology. So let's give you a quick definition. I hope that most of you understand what that word means. But homology means structures that are similar because they are derived from a common ancestral structure. Okay? So are bird wings, bat wings, and pterosaur wings homologous? We'll come back to that later in the semester, but it is a deceptively complex answer to that question. But it's very, very clear that all of these wings are derived from the forelimb of some ancestor. Okay? Now, despite the similarities, because they all come from a forelimb, there are distinct differences. So a bird has feathers and a rather reduced bone structure, and the feathers provide the flight surface for their wing. In bats, they don't have feathers, they have skin that is stretched between their 5 digits. They still have all 5 of their digits. You can see the thumb going all the way back to there's the bat's pinky finger with the skin stretched between them and the hind legs. And that provides the flying structure. So in function, very similar to bird wings, but the way it's built is quite different, particularly when we look at the fine details. And then finally, the pterosaur, which also has membranous wings, doesn't have feathers, but it's very different from the bat wing. In the pterosaurs, the 4th digit is greatly elongated. So that's the 4th digit, basically the equivalent of your ring finger. And from that, a reinforced 4th digit, we have this stretched membrane that stretches all the way and attaches to the lateral side of the body here. So again, a little bit more similar to the bat wing, but still quite distinct, particularly at the level of the bones. Okay, so fairly simple answer then, right? Where did these wings came from? The bird wing, shown here with the bones, the bat wing, and the pterosaur wing all came from a common vertebrate forelimb. And we have a forelimb also. So the human forelimb looks similar in very general terms to all of these, right? So, and I could have put a dog here or a turtle or any other tetrapod vertebrate. But we've got a humerus, right? Birds have a humerus, bats have a humerus, pterodactyls have a humerus. A radius and an ulna. There's the radius, there's the ulna. Same thing in the bat and the pterodactyl. So despite differences in shape and size, we have many of the same ancestral bones. Now, evolution has changed the organization of the wrists of these. So wrists are different. So this is an elongated wrist bone in the pterodactyl with a few there. A little bit, the bat has more numerous small bones, something's a little bit more similar to humans, which makes sense, right? Because we are both mammals, so we would expect us to perhaps share more similarity to the bat than we would to the bird and the pterodactyl. And then finally, digits with phalanges, much the same number. Birds have reduced number of phalanges, actually only have 3 phalanges remaining. I'm sorry, 2 phalanges and a pseudo phalanges here, which is actually, I think, a modified wrist bone. Pterosaurs have 4 of them. They've lost 1 of them. Bats still have all 5 of their digits, which is the ancestral form. And although humans have a modified forelimb, it's modified in a very different manner and adapted to different functions than these, which show some superficial similarity because they have all evolved to fulfill a similar function. Now, anytime you see a tetrapod vertebrate, it's very clear you can homologize limbs fairly easily. Some tetrapod vertebrates have lost limbs. So whales have lost their hind limbs. Manatees also. So they're the only mammals that have lost limbs, but they maintain their forelimbs. But in other groups, they've lost both hind and forelimbs. There's some amphibians that have done that, snakes, and some groups of lizards are very obvious. But if they have forelimbs, it's fairly easy to homologize them. So I'm going to put another diagram up here, and I'd like you to tell me from a biological point of view what's wrong with this picture. All right, so Pegasus, right? So if you look at this, and if you were to then compare Pegasus to, of course, horses would probably be the first thing you would go to, but any other tetrapod vertebrate, you would notice something immediately problematic, right? In that all other tetrapod vertebrates have 4 or reduced if they've lost it. They might have 2 or no legs, but there's no such thing as a 6-legged tetrapod vertebrates. So these wings in this, of course, fictional animal, right? This is a mythical beast. This does not exist in nature, and you would probably, even if you didn't know much about it, if you knew something about the history and the diversity of other tetrapod vertebrates, you very clearly say, no, this isn't right. Even if you'd never seen a bird before and you didn't recognize these as bird-like wings, you'd say, wait, where would these come from? This is just not a reasonable thing. And dragons the same way. Most dragons are tetrapod vertebrates with just bat wings that are kind of pasted on top. Right, so there are problems. Some dragons in different mythologies have four limbs that are modified into wings, and that would make a little bit more sense, and maybe something similar to a pterosaur or a bat, right? But of course, dragons are fictional beasts, probably originally dinosaur bones or other large fossilized remains that were mistaken for a large reptilian flying organism. But again, dragons like this in mythology just don't exist. But you would know that immediately because you cannot homologize this third pair of appendages, right, the wings, to anything that we see in nature. However, you can do that with real wings, right? Birds, bats, pterodactyls, you can homologize them very easily and find their linkage to this common structure in humans and other tetrapod vertebrates. Now, for EvoDevo, then, we would want to begin to evaluate the genes that are being used to build the bird wing, the bat wing, the pterodactyl wing, and maybe even forelimbs in general, and say, is there a core set of genes that are common for forelimb development? Are there new genes that have evolved in the birds or the bats or the pterodactyls to build wings? Or are they old genes that were present in ancestors that have taken on a new function? And so we'll begin to look at many examples, not just wings, but we'll begin to look at these specific examples and the research that we have been able to complete to determine and begin to answer these questions. How do we get DNA to turn into these wonderfully similar and yet diverse structures? Now, let's go to our fourth example of the origin of wings among the animals. And this, of course, is the insects. There are many different groups of insects that can fly, but they are all evolved from a single flying ancestral insect. And there are some insects alive today that branched off from the other insects prior to the origin of wings. And so we have things like, this is commonly called the silverfish. It's not a fish, but it's a type of an insect that is a branch of insects that diverged from all the other insects before wings evolved. Okay, so again, we can begin to say, okay, where did these wings come from? Let's begin to homologize. And so just like we might take a mammal and compare it to the bat and make a distinction about where bats' wings come from, we could maybe do that with insects. But it becomes a little bit more problematic. So notice that insect wings are actually a lot, when we compare them to the non-flying insects, whether fossils or the living relatives of the earlier non-flying insects, when we make these comparisons and try to make homology comparisons, we have 6 pair of legs in the non-flying insect and 6 pair of legs in the flying insects. We have a head and then a 3-segmented thorax. We have a head and a 3-segmented thorax. And there is no clear structure from which insect wings are derived. And for many years, this remained a great mystery. It's beginning to be answered, and there's still a couple of hypotheses. We'll, again, save the details of that for a later discussion. But be aware that insect wings are not a very easy, simple connection between other living organisms and their structures that may correspond to insect wings. So insect wings are a little bit more like the Pegasus or the dragon in that there is no clear predecessor for where these wings came from. But of course, a real example, not a mythical one. Now, in the last 30 to 40 years, there have been many great advances that have occurred in EvoDevo. We're going to talk about just a couple. This is just to give you a little bit of a preview of things to come. So one of the very earliest sets of experiments in the early days of EvoDevo were with fruit flies and looking at mutants. and finding rare mutants that had these really unique, distinct things. We're not talking about a mutation that made something completely that looked like a tumor or looked like nothing else they'd ever seen before. This is a fruit fly mutant that has leg-like structures growing in place of its antenna. And so these were called domain or homeotic mutations. And so these homeotic mutations begin to give us clues about the genes that we should begin to investigate if we want to look at the diversification of appendages. And so really this was kind of these early fruit fly mutants were some of the first steps towards making that link between morphology and genes. And of course today we have many diverse and quite advanced genetic tools that we can use to begin to make these connections even more detailed and understand them even better. One of which is being able to visualize the expression of genes. We'll talk about gene expression in our next discussion also. So if that term's a little uncertain in your head, put a pin in it. Hopefully you know what it means. But if not, that's okay. And we'll go over it in a bit. So we can actually add fluorescent molecules to genes as they are being used and determine what genes are being used in different parts of the body during the early stages of development. This is a fruit fly embryo with the head here going all the way back kind of in like a fetal position to the tip of the abdomen right there, and looking at 7 different genes and where they are expressed and the different parts of the body that they are responsible for helping to establish. These are all homeotic genes, sometimes commonly called HOX, and we're going to spend quite a bit of time in this class looking at the HOX genes in more detail. But by being able to visualize where those genes are expressed, we begin to get clues as to their function. And then finally, we have many tools. RNAi was one that's still used more widely in the past, that allow us to do functional assays. So not only can we just see where genes are being used, we can begin to manipulate the expression of those genes and turn that expression on so we get more of a certain gene product, or turn that expression down or maybe even completely off, and see what the morphological consequence is of that upregulation or downregulation in gene expression. This is a double homeotic mutant of a beetle. So the wild-type beetle larva has a head, a thorax with a leg on each segment, and then an abdomen without any appendages at all. But if we suppress the expression of 2 genes, UBX and abdominal A, then we begin to grow legs on abdomen segments where legs naturally never would form. So this gives us a clue about the past, the evolutionary history of this species, and also about the genes that are important as we begin to transition away from whatever the ancestor looked like towards our modern insects. And so sometimes our evolutionary history can be unraveled by changing expression and looking at stepwise expression, the evolution of expression in organisms. So it's a really rich and valuable set of experiments and set of research tools and model organisms that will allow us to more fully elucidate this connection between genetics and morphology.